I'd like to focus a little bit on the current status of personalized therapies and the approaches we're taking to try and make some headway in better drugs that are tailored to patients. And I think the motivation for this, there's several. One of the motivations has to do with the process of drug discovery, and the paradigms that have been used, they're outdated and clearly not working. And by the time we find a drug that can be brought to market, the amount of money we have spent developing the drug makes it inaccessible to most people. And even when it is accessible, it doesn't work for most people. So for the top 10 grossing drugs, if you look at the response rates, for every patient who responds, there's between 4 to 20 patients who do not respond. And a lot of that has to do with our notion that we're all representative of the population average when we're not, when we're all unique. And what we really have to figure out is what is unique about us at a molecular level in a way that will allow us to personalize therapies. And this is what I'll be speaking about is how do we figure out the unique molecular and network dysfunction within an individual patient. Where do you begin? The whole notion of N of 1 experiments, while exciting, I think is a bit misleading because there is no statistical power when you look at one patient. So the idea of looking at a cohort of patients, uncovering all the mechanisms that can go awry to cause a disease, and then unravel the molecular dysfunction is one part. The second part is to map a patient into those networks so you can uniquely identify which networks have become dysfunctional in that patient, and thereby tailor therapies that are going to be uniquely matched to those characteristics. And even then, I think we will make significant headway, but I don't think we will get rid of issues of recurrence and resistance and so on, because that has to do with the heterogeneity within the tumors. Since we're speaking about cancers here, every tumor is a mass of heterogeneous tissue, and even if you were successful to wipe out 99% of the cells, there will be a small fraction that will remain refractory, either because they're phenotypically tolerant or genetically resistant to the drug. And there's all kinds of theories coming up about how that happens. But those cells then take over and come back even more aggressive. So what we have to do is figure out how to stratify patients more accurately. And the general notion is when you have a group of patients, among them are subgroups that have similar mechanistic dysfunction responsible for the disease manifestation. And the challenge is to now use these modern technologies for profiling. So you can profile genomes, epigenomes, transcriptomes, proteomes, you can do all kinds of omes. And you have large amounts of data. And then what you have to do is figure out from those data which patients can be subgrouped into distinct subtypes of disease, and do that in a data-driven way. So you can even rank order the patients based on the progression or the severity of the disease, and then subtype them. Now this is harder to do than said, because when you think about systems, and the title of the talk is Systems Biology, and the reason we call it a system is because it's made up of many individual components, where none of the individual components have the properties that manifest in the entire system, but collectively they give rise to these emergent phenomena, which we call phenotype. And so when there's dysfunction, even in one component, the consequence of that tends to reverberate across the entire system. And you have changes that are happening in secondary, tertiary aspects of the system that are removed from the primary dysfunction. And so when we make these large profiling exercises, I think we end up with these very large amounts of data, and we're just piling stack over the haystack we already have, and the needle just becomes harder to find. And the question is, how do you then find in this vast amount of data that one little characteristic that is telling of why the person has a disease, one or few things? Now, the approach people have taken it's not new, but it can be misleading. The idea that if you see change in one feature and you find a correlated change in another feature, that correlation implies some kind of functional association. It is a very powerful way of finding these connections in these systems, but it's misleading, and it's difficult to emphasize how misleading it can be with these data. So let me just, if you indulge me, take some other data sets where you know what the features are, so you can appreciate what some correlations might really mean. So these are data from the CDC and USDA and the real data sets that were mined for associations. It was done by a guy called Tyler Wiegand, and he's got a website devoted to this. And here are some of the correlations you find. You find that people who drowned after falling out of a fishing boat correlate with marriage rate in Kentucky. And you also find that age of Miss America correlates with murders by steam, hot vapors, and hot objects. And here's a third one. I think I've made my point already, but per capita cheese consumption correlates with number of people who died by becoming entangled in their bed sheets. And the obviously ridiculous correlations make no sense. And the reason you know they don't make sense is because you know what the two features that are associated mean. But now think about mining the metabolome and transcriptome and epigenome, where the features are cryptic and they have labels that don't make any sense, but they make beautiful graphs. And you look at those graphs and you decide to do experiments and find drugs and treat patients, and think about the risk you bear when you take that path. And as a basic scientist, I know you can spend months chasing down a rabbit hole. So you don't want to do that. So how do you go from correlation to causation to mechanism? So that is something we have to think about. And I'll give you some ideas about the way we do it. And the basic idea is network identification. So you have a system, any complex system. It has within it a complex set of interactions that happen dynamically to process information, to take input and convert it into an output. And if you take such a system, and we are such a system, an airplane is such a system, a cell phone is such a system, and if you were to perturb those systems and observe their outputs, and you had some basic understanding of the principles of interactions. In biology, we can think of how proteins interact, how genes are regulated, how metabolism occurs. There's some general biological principles that you can use as constraints. Then you can do network identification by prioritizing the types of associations that have biological meaning. And iteratively, you can find the networks that will help you then explain how the inputs were processed into the outputs you observed. And this sounds abstract, but it'll become clear as I give you examples. And if you do it in a predictive framework, meaning if you consider causality and mechanism, you can find networks that are not only predictive, but also actionable. And I'll tell you what that means. So here's an algorithm we developed many years ago to do this multi-clustering. And what this algorithm does is it navigates through all these complex data sets and looks for patterns that are supported by evidence across orthogonal types of data. And the theory here is that the likelihood of technical errors or even biological errors across different types of measurements are unlikely to be consistent across different technologies or labs that have generated the data. So they cancel out. What emerges are causal, meaningful associations. And so this algorithm it's a machine learning algorithm. It is finding these patterns, and on the top left, you're looking at genes that are changing in a coherent pattern in some patients to the left of that vertical dashed line. And you find those patients have coherent expression of these genes, but the other patients did not, so you throw them out of the cluster. But the patients are grouped together, and the genes are grouped together, not just based on those expression patterns. It's also based on some sequence signatures, maybe mutations, maybe elements within their chromosomes that have aberrations and so forth. And even regulatory mechanisms, that these genes are not correlated by chance, but there are putative mechanisms for some factors that are controlling them. And you can also find patients who are excluded because those mechanisms have become dysfunctional in those patients. So there's a number of ways you can use it. And this is the stratification part. So you stratified patients and processes based on certain features. This is step one. And you can do this in a highly parallelized manner and find thousands and thousands of signatures. And I'll give you examples, real examples from cancer data sets. But then the question is, what was dysfunctional in those patients? Then that's the network identification part, the causation mechanism part, where there's a series of different algorithms that we've used, and I'll give you a couple of examples. And the goal of these algorithms is to find that wiring diagram of the factors, the transcriptional factors, the microRNA factors, the epigenetic regulators, and so forth that are modulating the patterns that you've observed. And these patterns, once you observe them, and the wiring diagram has predictive value, and you can test it on new data sets by saying, if I were to input those values, I can recreate the behavior of genes in that particular signature. And the reason I'm going into this into so much detail is because you use these networks to then find therapies in patients. Okay, and I'll skip over this a little bit. And we showed this many years ago that this network is predictive over the entire set of genes, and we can do this for any organism, human, diatoms, tuberculosis, you name it. We've done it there. And here's an example of how you find the wiring diagram. So let's say you have a set of genes that have coherent behavior. Now the correlation in their behavior is because there's some mechanism that is controlling them in a similar fashion. The question is, what is it? And there's many factors that can do it. As you know, there's proteins that can bind to DNA and modulate gene expression. There's microRNAs that can bind to transcripts and control their stability and so forth. And here's an example where the algorithm looks for the active binding sites for a transcription factor, a protein called USF1, which can potentially bind to 3 locations because they have sequences that match its recognition sequence. But among those, using the ENCODE dataset, which is a dataset that has profiled accessibility of chromatin, we find that only 2 sites are accessible. And furthermore, when we zoom in down to nucleotide resolution, we find that only one of them has evidence across 200-odd cell types in human that a protein was protecting that location by binding to DNA over there. So by doing this layered exploration, we can say this protein binds only to this location, and we can confirm it using some other method. And this is the only active site. So this is a wiring diagram for that protein to that site. And we can do that for all the different genes that fall into a signature. And we can do the same for the microRNA side as well. And I'm going into too much detail, but you can look for sequence patterns that are conserved in the 3' untranslated region of the transcripts of genes that are co-expressed. Take that matrix of nucleotide conservation and then convert that to a hidden Markov model and find a cognate microRNA that is controlling. So we can use a number of different ways to do this, and we can even map mutations onto the network. So we built all of this. So this was the conceptual introduction, which I spent most time on. Now I'll quickly go through an example of how we applied it to glioblastoma, where we strung these algorithms together, and we can put into this all that complex profiles we get from patient cohorts, and then find maps like this, where we can say there are mutations that are perturbing certain players that control networks that eventually perturb hallmarks of cancer. And in glioblastoma, this is the map we uncovered, but a very quick introduction to the data set. So we had about 422 patient biopsies, and these are tumor tissues, and 9 postmortem control. Remember, there's a heterogeneous mass of tumor tissues from each patient. And the question is, these are cross-sectional data sets. Can you really find such patterns in completely different individuals? Does this approach have the power to do that? And we found about 1,800 signatures, of which about 500 were found to be disease-associated because they had to do with hallmarks of cancer, or they were predicting clinical outcome. And I'll give you an example of such a signature here, where you have a set of genes. I think there are about 16 genes in the signature. And remember, there's nearly 500 like this. And we've ordered the patient. Every dot here is a patient. And we've ordered the patients based on the median expression level of the genes in those patients. And then we painted onto that the 5 different subtypes of GBM or gliomas, and then we can find that as the trajectory goes up, the subtypes are stratified, not cleanly, but you can see the patterns. Now, either the trajectory is incorrect or the diagnosis that the subtypes are based on those diagnostics are inaccurate. So I think this is the right way of stratifying patients. It's data-driven, and that there might be some missed calls because we misdiagnosed the patients. And then we can ask, is this giving us some information about clinical outcome that is useful? So we can take patients who are from the lower quintile and patients from the upper quintile and ask if they have different clinical outcomes. And it shows that there's a distinct difference in the survival. When the genes are high, the survival is poor. And we can then look at the maps that are around these signatures that are clinically associated with the disease. And we can look at maps such as these, where you can say these mutations are perturbing the networks which control these genes that are associated with these hallmarks of cancer, like evading the immune system, invasiveness, proliferation, evading apoptosis, and so forth. And we can then think of drugging those networks. And here's the map of the whole map from the 422 patients. Looks very complex, but this map can be mined for meaningful information. And I'll give you one example, and the example will be this one over here, where we find that mutations in these two proteins, NF1 or PIK3CA, are predicted. So patients whose tumors have those mutations are predicted to have elevated expression of IRF1, and the network goes on to predict that this factor is controlling 27 genes that are associated with a signature that predicts clinical outcome. And there's a series of tests we can run to confirm that the flow of information from the mutation to the phenotype is actually causal and mechanistically correct. I won't go into that now in the interest of time. And the genes are associated with the antigen processing and presentation machinery for MHC class 1. And so based on that, we can predict that patients' tumors who have these mutations will have elevated or increased tumor lymphocyte infiltration. And so we can go and test that in tissues. And remember, we have a large network with hundreds of such hypotheses, and different patients have different combinations of these perturbations. So based on that, we can get a comprehensive molecular phenotyping of a patient's tumor. And we can then decide what drugs are likely to work based on those characteristics. And we can do things like this. For example, in patients who have, and this is actually based on the City of Hope trial, where the patient was given CAR T cells against IL-13 receptor alpha 2, but came back because of presumed suppressed expression of the receptor. So the question was, can we find the network governing that particular receptor and then stimulate expression of the receptor? And can we find molecules that can do that? And we're doing this with Mike Jensen at Seattle Children's. And so the prediction was when mutations in interferon gamma pathway are present, then you have this network that is perturbed, and that causes the suppression of IL-13 receptor alpha 2. We find all the mechanisms, and we predict that, okay, here's a drug, and this could be a repurposed drug, and this one was, that you can give to the patient to then disrupt this network so the receptor is expressed again. Or if you have a tumor tissue, you can treat with the drug, prime all the cells of the tumor so they're expressing the receptor, then give CAR T cells. And then we showed that at very low levels of this drug, which I can't tell you what it is right now, is you see elevated expression of the receptor. So this would be the idea of combination therapy, where you have a proven therapy that is effective, but does not give complete remission. There's recurrence, and so you can give a second drug to make it even better. And this is how the N of 1 trial idea would work, is when a patient comes in, we have this map already laid out. The doctor orders some profiling tests, and then the patient is mapped into different signatures based on those profiles. And based on where that patient maps, you can predict outcome depending on the patient information you have, clinical information you have on those patients. You can find the networks that are perturbed, confirm it with other profiles of the same patient tissue, and then run screens using patient-derived cell lines. Now, this is where I think the current drug discovery pipeline is broken, where we take a prediction and we go and we test it on a standard set of models, cell lines, and PDX mice. Now, slowly, more and more people are archiving tumor tissue, so you can go back to the same patient cells to test out some of the therapies to see which drugs are more likely to be effective. And then you can design a trial based on those characteristics. So you can then have trials that focus on patients that have certain characteristics matched to drugs that are predicted to be effective against those characteristics. And that's the N of 1 trial idea, is each patient is now uniquely matched to the drug, and the trial is based on that map that you created. And this is actually, I didn't finish my train of thought here, an example where we found that romidepsin and a microRNA mimic could have a synergistic effect on proliferation. And we showed that the drug romidepsin, which is very toxic, that you can lower its toxicity by titrating in the microRNA mimic and get a similar effect at much lower doses. So this could also help at lowering the undesired side effects of drugs that are known to be effective but are not something allowed in the clinic because of the bad effects on patients. So just coming back to what I mentioned early on is this idea that the TCGA has profiled over 32 different cancers, and they have very large amounts of data. They just published 27 papers. ISP contributed to 11 of those papers. They all came out in Cell Press journals. And that's essentially a map of all the types of associations and profiles that were discovered from tens of thousands of patients. We started with one of those data sets for glioblastoma. Now we've also done that for multiple myeloma. We're now doing it for breast cancer. We have to keep building those network maps and then start driving some of these strategies where you can. The problem with TCGA is you don't have access to the patient cell lines. You don't have access to clinical information of drug responsiveness. There's a lot of information that's missing, but it does serve as a good landscape where you can begin your next study. And that's what we're doing with the, well, this paper is published, but that's what we're doing with the Providence St. Joseph Health System. And we were just affiliated with them a couple of years ago. And the idea is to take these maps and drive projects that are clinically focused to find why the tumor environment responds differently in different patients, and how do we find characteristics that are predictive of responsiveness to therapy. How can you find blood-based biomarkers that are tied to the signatures I described? And these are multi-biomarker panels made up of metabolites, proteins, microRNA, circulating tumor cells, cell-free DNA. Which of those have the information you would need to non-invasively diagnose a patient and also monitor their response to therapy? Especially in GBM, that becomes really important to do things like that. And the third is, with this Charles Cobbs here, his lab is just downstairs, is to do that patient cell line derived screens where you can have a patient matched to a drug in a way that you can test in a very short time cycle to make sure that the drug is effective based on the cell line screens, so you can prescribe it to the patient. So this is just a summary of how we can go from these large omics data sets to N of 1 trials. And I won't spend more time describing that, but just thank a whole bunch of people and especially NIH for this work. So I think I have some more time to take questions now. Thank you.